Yeah, good morning, everyone. Thank you, James and the organisers and you guys for selecting me to give a talk. What I'd like to present is some of our recent research, which is on something which wasn't what I'm not a neuroscientist by training, but we accidentally discovered something which I think points to something really spectacular, which I'd like to share with you. And that's the discovery of molecules which have got memory. So these molecules can remember information in a binary format. And these protein molecules are actually what holds our brains together, leading to this idea that maybe there's a binary code in our brains. That sounds a bit far out there, especially first thing on a Wednesday morning, but I'd like to give you a bit of background to this and why I think it's true, and then hopefully have a discussion on what the implications of this would be if we can prove it. So in the middle of this screen, I don't know if I've got a laser pointer. No, maybe not. But in the middle of this screen, we've got this protein called talin. This is what we work on. And I'll show you a bit more detail on it now. But from the youngest child, from a kid who's just started, right through to the oldest adult, everybody knows where information's stored, where we do all our thinking. It occurs in our brains. And the electrochemical signaling which goes round and that brain activity, we all have an understanding of this. And if we looked at this baby's brain in detail, what we would see is millions and millions of neurons, which are the brain cells, all talking to each other by sending these electrochemical signals. And this is really complex. This is well established. But what's not so well established is where is the actual information in this? Is it just in the electrochemical signaling, or is there something underpinning where the information resides? And this is incredibly complex. We're nowhere near understanding these electrical circuits. But what we've discovered is that when we see these, where these neurons are talking to each other, they make these connections called synapses, where one neuron talks to another, and it can send signals across this tight junction here. These are always drawn like this, just floating in space, free and all, like two halves of a coconut. But in reality, that's not what's going on here. These are inextricably linked. And they're linked via this really complex scaffolding. So this is held together because there's a scaffolding both around it and inside it. And a bit similar to this lighthouse where this scaffolding is holding it while they do the repairs. But what we've found out is that scaffolding holding these synapses together is built of these memory molecules. It's built of these binary switches. That's what I would like to just present as an idea to you guys today, and hopefully leave time for questions, because it opens up a few interesting possibilities, I think. So the reason why that is, I'm going to cut straight to a video because this illustrates what I'm going to talk about, but then I'm going to talk about how we discovered this, is that these proteins, these scaffold proteins, are mechanically active. If you apply a small amount of force onto these, you can open these switches. So these switch domains open and close in response to mechanical signals. So that electrochemical signaling going around the brain is switching on tiny motor proteins inside all of our cells. And those motor proteins pull on these molecules. That's the idea I want to try and show you today, and how we got to that. But just to say that these are switches. They can have two states, either a folded or an unfolded state. So they've got two states, a 0 or a 1. This is binary information being written into the shape of this molecule. And it led to this idea. Once I was working on these memory molecules, and then as I started to look where they are and found that they're holding our brains together, every single synapse is held by these memory molecules. And if you delete the memory molecules, you don't have any synapses. None of the neurons talk to each other. So they're a fundamental part of these circuits in our brain. And it led to me writing this paper a couple of years ago now, in 2021. But I just want to go back 10 years because I didn't start as a neuroscientist. What we were interested in was, I don't know if it's going to play. There. We're interested in this. Does anyone want to have a guess what's going on in this picture? I don't know if you ask questions in these auditoriums. So this is two cells. These are fibroblasts. They're skin cells. They're what crawl under our skin. If any, hopefully you're all too young to have tattoos. But if you have a tattoo, these can be what takes up the colour. But this cell is, if the monitor was working properly, he's crawling along. Oh, blooming heck. is crawling along in a really ordered fashion. We wanted to understand this process. How can a cell crawl in such an organized fashion? And the reason it's crawling is because it's making these large assemblies which are in green. And this is where this cell physically grabs on to an underlying cargo net called an extracellular matrix. So all of our cells in our body, we're not just a massive pile of cells on the floor. We've got tissue, we've got organs, we're organized. And how that organization occurs is because our cells are connected to this extracellular matrix and to each other. And that's where the order comes from to give rise to an organism. And this is really, this cell is physically pulling itself along that cargo net. And in doing so, it's getting to where it needs to go. And why we were interested in this was because it's essential for the development of multicellular life. The adhesion molecules we're going to talk about occurred at the advent of multicellularity. But it also goes badly wrong in many different diseases. If you have a cell which suddenly becomes migratory and starts to crawl around the body, that's a really bad thing which can happen. So we're interested in understanding this. And just to give an example of that, this is computation we're talking about, and I mean in terms of like working stuff out and actually doing sums. There's a really beautiful experiment. Does anyone know what a stem cell is? Exactly, it's undifferentiated. It's got the capacity to turn into any other type of cell. That's a great answer. So here, this experiment is really very simple. So I'm not going to show you too much data, but I think this is important for trying to see that this is computation. So here we can take one of these stem cells. It's got the capacity to reprogram. Depending what signals it receives, it will reprogram, and it can do lots of different jobs and differentiate. And this stem cell can differentiate into a fat cell, or a nerve cell, a neuron, or a muscle cell, or a bone cell, depending on the signals it receives. But in this experiment, this was done by Dennis Disher's lab in America. This is a beautifully simple experiment because everything is identical, and the only thing they change is the stiffness of this substrate. If they put the cell onto jelly, it becomes a fat cell, and if they put the cell onto glass, it becomes a bone cell. And in between, you get the different types. So the only thing changing here is the physical parameters, the physical stiffness of this matrix. So this cell's grabbed on, it's feeling it like that, and that ultimately leads to switching on the correct gene expression profile to become a completely different cell. So this is mechanical computation, and this is what we work to understand. How does that cell get those instructions to do that, so fundamentally reprogram? And it leads to this notion that these small blue squares where this cell's grabbing on, those hands where it's pulling, is where this computation's occurring. It's grabbing on, and then it's using those attachments to learn what to do and to adjust. And if you look at those blue squares in a bit more detail, what you find is on the surface of every one of our cells, we have something called an integrin. These are receptors, and I don't want to go into too much detail about them, but they're essentially the hands of the cell. So each of our cells has got these hands which can grab on to this cargo net-like extracellular matrix. And in doing so, it can grab on, and then it can start to interrogate what it's attached to. But it can also use that grabbing on to stay where it is, or if you go ape or somewhere and you're climbing on the cargo net, for actually moving around by using this adhesion in a controlled fashion. So this is what we want to understand. And the memory molecules which we work on sit here just inside the cell. So on the edge of all of our cells, we've got receptors, and then just inside, we've got these proteins which have got this molecular and mechanical memory. I don't know if I did that. Did I turn that off? I don't think I did, because there's no... Oh, here we go. So, yeah, they've all got this attachment. They grab on, they can feel it. But those... what it would be showing now, it would be showing those hands of the cell connected to the memory molecules, which are then connected to the cell's cytoskeleton, which is like the musculature, actin and myosin. I don't know if you're familiar, but when we lift anything, we've got muscles in our arm. They're made of motor proteins and actin. And thank you. In our cells, we have exactly the same. Our cells have a musculature. They can make these filaments, and they can pull, they can push, and they can, if you imagine that cell moving along, it's literally pulling itself forwards. So this is where I think, as I'm going to show in a minute, this machinery, which is in those stem cells, is also the exact same machinery which is holding the synapses of our brain together. And this means that these molecules here are perfectly sensed to respond to forces, because if the outside world changes, they can detect it. But if the cell can be given instructions to switch on its motors, it can physically pull on these memory molecules and change their shape. But what's striking is this is 4 proteins, but they do many, many different jobs. So this computation is fundamental in all of our cells. So we already know that this is a migrating cell. Does anyone want to have a guess what's going on in this cell? In this cell, it's not a cell actually, but anyone want to have a guess what's going on in this bottom corner? Yes. It is moving. So this is the musculature, the muscles of a Drosophila, a fruit fly. It's what, say? It's not elastication. This is literally the muscles in a fly. So you guys are hopefully too young to drink white wine. If you drink white wine in the summer, you'll almost certainly get a tiny little black fly in there. That's called a fruit fly. And they're incredibly powerful genetic models. And this is a fruit fly. It's an embryo of a fruit fly. It's just becoming contractile, so it's just clenching its muscles for the first time. But all this yellow streak is this same talin, integrin, and actin holding this together. Oh. And just, oh man. Right. Does anyone know what's going on in this bottom corner? I heard it over there somewhere. It is mitosis, exactly right. And if you look what's going on in it, if it's going to play, the cell starts off spread, it's grabbing on, but if you want to divide into 2, if you're spread out really thin like a pancake, it's really hard to divide. So what the cell does is it lets go of all of its adhesions just as it's about to go. It goes into a ball, and then it splits into 2, and then it immediately spreads out again. So this machinery, this computational machinery, is playing a pivotal role in all of these jobs. But what we're most interested in is these connections in the brain. And I don't want to go into too much detail on the structure, but I want to just point to, this has been a long-going project. I solved this in 2013 with a big team when I was at Leicester. And on the back of this, I set up my own research group to work on this molecule. But what happens is it grabs on at one end, and it's got, all I want you to draw attention to is these 13 rod domains, they're called. These are bundles. And what we found 10 years ago was we identified that one of these was behaving like a switch. And by that, it was doing one job when it was closed, then you pulled on it and it changed. So what we've been doing over the last couple of years is trying to make videos to animate this, because I'm telling you something quite complicated, but a video is much more pictorial. So here's our talin molecule, and it's getting pulled at this end. But what's happening in this video is that before you pull it, it's doing one set of jobs bound to this yellow protein. Hopefully it'll go again, here. But when we apply a mechanical signal, we pull, we change the binary switch, and in doing so, we change what it's doing. So the whole cell reprograms as a function of these mechanical cues. So if you could switch on the motor proteins inside of a cell, you can update the status, you can write information and change the shape of these molecules. And what I would set up my group to do was to understand how much force, how hard is the cell pulling. If it has to pull really hard, then it wouldn't ever happen. And if it happened without even pulling, it would be bad. Just as a, oh man, sorry. As a shout out, all of these videos are on my YouTube channel, and they were made with a brilliant scientist called Sam Barnett, who's just gone to work on the 1,000,000 Genomes Project, no, 100,000 Genomes Project. But as a big plug for science, and hopefully you guys are here for science, so this is one of the STEM's a hard subject, but it's also so rewarding. And one of the beautiful things is it's truly international. And we work here in Kent, obviously, but we collaborate with all of these different labs all around the world. So we're the world leaders in the biochemistry of these proteins, but then each of these is world leaders in what they do. So we work together to solve these complex problems, and what I'm hopefully going to show you today. But the next experiment was done in collaboration with the guys in Singapore. I go there every year to do these experiments. And this is the people who do it, Ming Chi and Ji, absolutely amazing scientists. They're biophysicists. And if you want to know how hard you need to pull on a single molecule, it's quite complicated because it's really, really small. But what these guys have developed is they've developed a technique to pull on individual molecules. And how you do it is you make a protein. So this is one protein. So anyone who remembers Avogadro's number, well, I don't know what it is, 10 to the 23. This is a single protein. But we attach it to a microscope via one end, and we attach a magnetic bead to the other end. And then we can pull on that using forces similar to what the cell can generate and watch what the protein does. And when we do that, you're pulling on, here we're just pulling on 3 switches. When you pull it, it gets stretched taut. But then you see these big jumps in extension. This is where these switches have opened. So here's switch 1 opening, switch 2 opening, switch 3 opening. And then you let go and it immediately resets and they close. So this is 6 cycles of opening and closing these switches. So this showed us for the first time that these are true binary switches in these proteins. And if you look at what the whole molecule looks like, this is where this gets really complicated. This is pulling 13 of these switches. And it's the same experiment where each of the, we're pulling on them, and as we increase the force, so anyone who does physics, they'll be working with pushing a weight across the table, or if you're lucky, you put it on wheels and put it down a ramp. But you're looking at Newton-level forces. Here we're looking at minuscule piconewton forces, which are what the cell can generate. So it can get an instruction, it can generate a piconewton level of force. But each of these steps in this is one of these switches opening up. So this molecule is very, very it's a series of switches, and it's very changing its shape dramatically. But because each of these switches is binding to different proteins and different parts of signaling pathways, what's actually happening and what we're working on in the lab is that each of these steps is switching on and off different cellular programs. And this is this binary coding which we're interested in. And this is what I think is fundamental to all animal life. And this leads to this notion that this is memory that we're talking about, because each time we pull on this, we can update it by opening one. This thing's not happy at all. I haven't even got a keyboard to press skip on. Next slide, please. I was going to say on the COVID things. Yeah. So each time we pull on this, we update this register, we update this string, this binary string, in a way controlled by the cell's signaling. And if we pull, we open these, and if we relax slightly, we can close these. So this binary string is constantly changing and writing information into the shape of the molecule. And where this is exciting, why we're now full-blown neuroscientists, is that we're very interested because the synapses are held together by this complex connections between the pre and the postsynaptic side and the astrocytes and the glial cells. But this is held together via these memory molecules. These talins, which are holding this together, are around the edge of every single synapse. So they considered to be purely scaffolding. We now can show that these scaffolds have got these switches in them. And then what leads to this idea that when you stimulate a synapse, what's been shown by other researchers is that the motor proteins in the stimulated synapse gets switched on. It pulls on these switches and updates the pattern of this, these ones and zeros. So this is truly binary information in the synapses. And so the idea we're trying to develop is that information is written into the shape of these molecules in like actually how we write information to a computer disk. One of the things that requires is it requires that the brain is a mechanically perfect system, because you're trying to do these tiny little forces. And for a long time, as a mechanobiologist, it was considered that maybe the brain's not good for mechanobiology because it's a bit soft and it's a bit squidgy. But what that actually means is it's perfect because every single one of these synapses can build its own little environment, perfectly isolated from all the others, to enable this signaling and this mechanical computation to occur perfectly. So we're working with to actually look at this in brain slices and in different disease states to actually read the shape of these molecules within samples of neurons. And that's the big idea behind the MeshCODE theory. We've got this binary coding. We know that it's in every single synapse, and we know that the electrochemical signaling switches on the motor proteins. So the idea here is that this signaling changes the state of these switches, and then those switches change the activity of the synapse. I'll skip through because I want to get to the question. So one of the things I really want to show is that like this is what a synapse looks like, and this is something we're actually doing at the minute. we're physically measuring where the talin molecules are, where one end of the talin is and the other, and we're watching these molecules change shape within the synapses when we stimulate them. So here, I don't know if you can see it very well, but these green and these pink dots, this is the two ends of a talin molecule within a dendritic spine. So there's 4 spines on this page, 4 synapses, and each talin is in a different length. And that's shown as a cartoon here, that these different lengths of the talin are spanning this whole thing. But what they're doing is they're positioning all the enzymes inside that isolated synaptic unit and organizing them. So this is what we're measuring now, and this is what we really want to link to. We want to show that these lengths correlate to the activity and that this is information written in there. But it leads to a model where if you've got 2 bits of the machinery, one which is static and one which you're moving, you can set different activation states. So you could imagine that by organizing these enzymes differently, you could have 4 of these spines on a single dendrite, but each one of them could have the enzymes organized differently as a function of this binary coding, such that they have different activation states, even though if we look with a microscope, they all look exactly the same. And you might just be starting to learn to drive now, but in the UK this works really nicely. Overseas, in America particularly, they've never heard of a crankshaft or whatever they call it, but we've got manual gears in this country by and large. And how that works is that you use a stick to apply a force to move a mechanically operated part of the machinery. Does that mean we have to run out? No. Relative to a static part, and in doing so you put this into different settings and that gives different amounts of transmission, exactly the same as what I think is going on here. You put this synapse into different settings and then you have different amounts of activity, and then as the signals go through, it'd be like changing the gear on that synapse. Each one would get dialed up and down. So if you could get on board with that you could write information into the synapse. If you have a really complex circuit of synapses, then there's an opportunity to write information in a binary format into these circuits. And if you look at what the cortex of our brain looks like, where information's thought to reside, it has this incredibly boring repetitive structure, where this is just I think we need a new battery on this thing for the next guy. This is 3 of these cortical columns. So the cortex of our brain is made up of 2,000 of these really repetitive units. But each of these red teardrops is a pyramidal cell. Each of these is making up to 60,000 synapses with its neighboring cells. And all of this is connected. I'm not even passing this. All of this is connected to the hippocampus via this trunk wiring. Where this is exciting is if you look at what we've done over the last 20 years in designing ways to store information in silico, then the optimal way, so you can have a gigabyte or probably even a terabyte on a disk the size of your fingernail, is that those SD cards, they have an arrangement which is, again, is incredibly repetitive. It just repeats the same channel structure with these same flash and pages and blocks, but gives them all a different number. And then you just have an addressable. You say that information's on channel 1, flash 2, or whatever. And then you can call it back just by having an addressable memory. And that's what I think might be going on here. I think that all the information might be written in the shape of these molecules in this cortex in a way that's mapped by the hippocampus, and we call it. So if I mentioned a kids program you watched when you were 6, you'll immediately recall it back, but you're not thinking about it all the time, hopefully. But that information's stored, and then you can just retrieve it instantly, within a second, and you call it back. Exactly the same with the disk. And I had to get my son to help me with this, because my maths isn't so good, but he's doing A-level maths. So he worked out with the number of switches in the number of synapses, then there's a potential for 10 petabytes of data to be stored in this binary format within this cortical region. But it leads to some really interesting discussions, because here's a guy who's about to go for a shower. It's put his hand in. He's looking to see whether it's freezing cold, he'll come out. If it's really hot, he'll come out. But he's making a decision on how much time he's got, how hot it is, whether he could tolerate that, how cold it is. And what's happening is the sensory neurons in his fingers are being stimulated, and they send these binary pulses, these action potential spike trains. So like pulses of electricity, like down these, what are essentially wires, through these synapses in the spinal cord and up to the brain to do the computation. But what's happened there is that this guy has received information and then updated himself or herself as a function of that. And this is just someone having a shower, but you could imagine taking this one step further, where every single thing that happens to you will be picked up via some sensory input in your eyes, your nose, your fingers, and fed into a computation. And I like to imagine that you could predict having an entire mathematical representation of that animal written into these switches as a function of every single stimuli it receives. And this would optimize it for its own unique life. We're lucky we've been born in where we are. We've got a very nice existence. If we were born in the North Pole or something, we'd have to get used to very different living. So you optimize with all this information written into this coding, optimizing these circuits. Is this going to work? So just as a few thoughts, then hopefully I've convinced you that we've got memory molecules and that they're scaffolding our brains. It leads to this idea that they could be written to, and perhaps each synapse in our brain can be specifically addressable by neural activity. You'd imagine that would be true. With a computer, we can address every single part. However, if that is the case, and you've got a petabyte of data, then it becomes really challenging to store and recall that information. So you must have some form of file allocation table. This is computer talk. But it doesn't matter whether you think the MeshCODE theory is right or you've got another pet theory of memory. Whatever your theory of memory is, you've got to have some form of file allocation table where you've mapped out where that information is stored in your brain so that you can call it back when you need it. So this is, I think this is the hippocampus, and I think this is generally, that the hippocampus is where the thinking, like the central computer is, and then it calls to these different parts. And there's a huge amount of research to understand this. Like this is like with EEGs, where you put the cap on your head and you read the electrical fingerprint. Like you can put one of those, a little tiny electrical cap, on a mouse's head, and then you can watch it go about its daily work. And this is work done by this Dragoon guy. But then you can measure the electrical fingerprint of what it learned in that day. And if you looked at it, it'd be spread over lots and lots of neurons and lots of regions of the brain. But once it goes to sleep, there's this process called memory consolidation. And that's where it goes to sleep and it pulls out and it builds that information into the rest of the information. And the next day you look at that, the fingerprint, the electrical fingerprint, and the synapses which are holding that information are streamlined and reduced. And if you look at this and you think of this from a data model of using electrochemical signaling to switch on motor proteins to pull on memory molecules to update the binary code, then this looks exactly like how you do data management if you're running a database. So this is off Wikipedia. This is the sleep cycle, when you go to sleep at night. And in the context of data management, this looks exactly like how you would build a database or rebuild it at night. And when you first start, if you're going to rebuild a database, pull all the information out and build it back in, you don't do that in the middle of the day where everyone in your company is using it, because it will get really corrupted. You wait until there's hardly any action on it and it's not being used that heavily, and then you can start to move and manipulate that data. So what happens when you first go to sleep is that you go into very deep sleep, you let those calculations finish, but then you go through these cycles of what's called REM sleep, where there's a huge amount of electrochemical activity, and it looks like it's just like going almost like haywire. But in this model, that would be altering these switches, changing the route through the neural circuit by setting the activity of each of these synapses. And then you would go back into another period of deep sleep while you let that calculations finish, and then you do it again, and you'd have 5 cycles of this. So maybe you guys are all too young to have done an all-nighter, but if you stay up working all night or going out all night, then the next day you're not working at optimum speed. And that's because you've not had time to process and sort all of that data, so your brain kind of gets fragmented. But the idea here is that if you get 5 cycles of this, then you can build what you've learned in that day into the structures and into the mappings of your thoughts in a way that then the next day it's just part of your lived experience. And then this is, this is data management, I think. I think that's why we have sleep. And I also think that's why sleep's essential. All animals sleep, and I think it is to enable this data management to occur. I think I'm out of time. But just to wrap it up, this sounds a bit crazy, but it came from an understanding and a discovery of molecules which have got memory, molecular memory, written into the shape of these switches. Experimentally, we can write strings of information into these molecules by using these tweezers. We can actually control that and write distinct patterns. We can also read the shape of those molecules because we can use the things which bind to them. We can make fluorescent versions and read what shape that molecule's in by what proteins are stuck to it. And what I think is, but this is based on biophysical principles. It's not based on any quantumness. It's not based on anything we don't yet understand. It's pure physics, just operating switches in a highly ordered structure. And I think it's completely consistent with all the other models where electrochemical signaling and the brain activity, which everyone knows, apart from here, it'd be updating where that electrochemical signaling is going, which synapses are active, which ones are not, in a way that encodes information. And I think what's important is that this identifies a read-write memory system. Again, it doesn't matter what theory of memory you is your favorite. Any form of complex computation needs a read-write memory system. You need to be able to hold a calculation while you do another calculation and put them together. You can't manage without a read-write memory system in any form of computation, which as we know it. And I think altogether then that supports the view that this is an organic supercomputer, which we are using computer logic rules, which we're also converging on our understanding of computers. So I want to finish there, and hopefully I'm not massively over time. In particular, I want to thank Ji and Ming Chi in Singapore. We started this just as a curiosity, how hard to pull that switch, and it just opened up this whole vista, and every experiment we've done and the papers which we're currently writing, it's just totally transformed how we see it, and I think we're transforming how all of this is seen. I'd like to thank Sam for the nice videos which he helped me make, and the people who fund this research, including the Human Frontiers, and the guys in the lab, and thank you all for listening. Hey.
Why do you think that talent's the most important memory story model?
So that's an absolutely brilliant question. So I think that's why I called it a mesh code rather than just a talin code, because talin's the only one which has got 13 switches in series, which is coupled in all synapses. But there are other mechanical switches in other proteins, but normally only just one or two. So the talin is the major one, but it binds to something called vinculin, which I showed at the start. It was actually the green in that cell which was crawling along. The greenness was because of vinculin. It's got 6 switches as well, so talin and vinculin is what I think is. But yeah, there are other memory molecules are available, but they just haven't got this complex switch pattern. Good question, though. Yeah, I hope I'm not around when we do, but yeah, I think so, because I think, like there's a talk like with a C. elegans worm, which is 1 millimeter long. They've only got like 400 or 500 neurons. So you'd imagine that at some point we'll be able to fully simulate one of those because they've only got like 100,000 synapses or something. At the minute, we're still establishing the rules, and we're trying to, as I showed, we're trying to see these shape changes in neurons in culture. We're trying to get this, now the full connectomics of a worm is mapped, so we're trying to look for this in a worm to see it, but yeah, I'd imagine so. And also maybe you could read this out. The big thing we don't know at the minute, which I don't go anywhere near here, is we don't know the operating system of a human. We don't know the format the data's written in. So there's still a long way to go, but yeah, I think we could read it. And then there's a really amazing bit of science fiction on uploading and the first uploading of a person, and the implications and the ethics of it. So yeah, I think possibly, yeah. Hey. Yes.
They're caused by like build-ups and plaques, like amyloid plaques. If you've got, are they like interfering with the kind of communication across the synapse adhesion?
Yes. So that's an absolutely wonderful question. So yeah, definitely the neurodegeneration is one of our main focuses, because the main cause of the amyloid plaques is a protein called amyloid precursor protein that binds directly onto talin. And one idea we're working on is that by the time you can see the plaques, the information was lost quite a long time before. The plaques are like a signature that it's happened, but maybe the information's getting scrambled and lost. Because if this is a coding between neurons, if some of them start to get out of sync, it's like getting corrupted, the data's getting lost. So we're working on the idea that the information's getting lost, and then you start to see the plaques. So we're hoping that we might be able to, if you can start to see it forming, to actually reduce that stage and slow Alzheimer's down by 20 or 30 years. If you can slow it down by 30 years, then most people would have died by then. So yeah, but absolutely brilliant question. Hey.
Do you think that improving our understanding of how the brain stores memory will improve our ability to store
Yeah, I hope so. So we use this, like the very common and popular in computing is neural networks, but they're they're very artificial, in fact, because they involve looking from above and not actually a proper true neural net. One of the things we're interested in is a quantized neural net where each node and each coupling has got discrete states. And then if you could have that and get the computation to work, then yeah, you could build much better computation, because here it's not just that neuron talking to that neuron, it's that neuron talking to that neuron, but it's getting input from other neurons, and then that gives the overall thing. So the amount of computation happening in a single neuron is really, really complicated, and then it's multiplexed by loads of neurons. So if we could build more information into each node of each connection in a neural net, then I think, yeah, definitely. Hey. Sorry, one more time with the microphone.
What can this help with? Like, can it help like AI or something? I don't know.
I don't know about with AI straight away, but possibly in 50 years. I think with the computation and the rules of computation, I think we'll hopefully make some progress on that. I'd also, I know I've finished, but one other thing which we're very interested in is that there's a type of child. I got contacted by Boston Children's Hospital because they've got a series of children who have got epilepsy, and it's all caused by mutations in the talin protein. So the idea is that, and they're all in where you pull on the protein. So the idea is that epilepsy might be caused because these synapses get out of sync because they're not pulled correctly. So we're really trying to hope that with Alzheimer's and epilepsy, we might find new ways to treat them as mechanical diseases. Hi. No, it's good. Both, I think. So with epilepsy, you can normally tell where the foci is because it's stiffer. You can see the ECM is stiffened. So maybe they're getting bashed is stiffening that. Something else which is interesting is if you knock yourself out, which hopefully you haven't, you lose the last half an hour, but anything before that you kind of have. So there's obviously a time dependence on how this is written. The very short-term memory, if you get a knock, then it gets scrambled and you lose that. And the same if you rub your eyes, you can see nice fireworks. It's like nice to do in exams when you're stuck. Then you see fireworks, you're mechanically stimulating those optic nerves and you're seeing it. So I think with the rugby players, yes, sustained banging and getting inflammation, which leads to a stiffening, which could then trigger the neurodegeneration for sure, yeah. Okay, so.
Let's see.
Yeah.
Is there any evidence that if a talin is destroyed, there's any redundancy within the synapse so that
This has been one of the best question and answers. Yeah, I think so, definitely. And that leads to an interesting thought process. It's actually by Francis Crick, who said it's hard to imagine memory stored in molecules because if the molecule gets turned over, then that information would be lost. So the idea we're working on is that the talin molecules are part of this machinery which is moving back and forwards. But you could also imagine in the brain, we're not just writing to one bit. We might have like some sort of RAID style thing where we write it almost like back it up or something, because there's bits where you lose damage to your brain, but then you can reconfigure some of that information. So it's all to be worked out. But yeah, I would predict that there is redundancy.
Thank you all so much. Firstly, you've got a gift for Professor Goult here.
Oh,
Say
wow,
thank you from the journal. Absolutely fantastic talk and some fantastic questions.
It really was.
As well. Yeah, really, really interesting.
Yeah.
So thank you.
Thank you so much.
Together. So thanks so much.